Dr. Mike Zinka. So Mike is the Executive Vice President, Head of Research and Development at Repare. Prior to joining Repare, he was the lead oncology bioscience organization at AstraZeneca R&D in Boston. In this role, he served on the Global Science Leadership Team, Oncology Research Board, and the Research and Early Development teams. And he was accountable for strategy and key collaboration partnership and delivery of an innovative portfolio of patient-centric drug discovery programs. He helped to transform AstraZeneca pipeline through strategic building of the immuno-oncology, tumor drivers, and resistance and DNA damage response areas. His group delivered multiple novel therapies in the clinic, including inhibitors of protein-protein interaction, enzymatic function, bromodomain interaction, and nucleic acid-based therapeutics in collaboration with Ionis Pharmaceutical. Dr. Zinda holds a BSc in biology from the Minnesota State University and a PhD in molecular biology from Vanderbilt. And he received his postdoc training at Princeton University and Eli Lilly Company. So Mike, thank you for joining us. You should be able to share your screen now.
Thank you. All right. Is everybody able to see that as a single slide?
Yeah, it works. Yeah.
Awesome. Thanks. I wanted to just, again, thank the organizers for inviting me to talk here at the PRiME Symposium and really to tell everyone a little bit more about Repare Therapeutics in particular. Again, as Tom had highlighted, newly IPO'd company, we do have to put a disclaimer up here about forward-looking statements. But then I'll just move on quickly to get to the key points. So one of the things I just wanted to step back and highlight up front was, the team, right? So we built together a very, I think, robust team starting in early 2016. But this really started with our key scientific founders, Dan DeRoche and Frank Zakary at the University of Toronto in the Lunenfeld, and Aniel Sphere at NYU. And they really formed that basis for the start of this company. Dan, with his deep knowledge in genome instability, DNA repair, as well as bringing in a CRISPR synthetic lethal platform that we've helped build and make more robust within Repare. Aniel Sphere, who had pioneered the pol theta area, a key target in the DNA damage response space, and really allowed us to start with a key target in mind. And then Frank Zakary, who is a key structural biologist, that's really helped enable our drug discovery efforts as we've continued to move forward. But this was in 2016, and we've now built up from seed funding all the way through different Series A and B financing to an initial public offering on the NASDAQ this year, in June. And have grown from that 1 to 2 people in that early seed funding now to over 75. So what was it that we sent out to do when we were starting Repare? And that was really to expand out the patients that we could treat through a precision oncology method beyond those that had been looked at for the last 20 years. So I think for those familiar with oncology, gain of function alterations like mutations in EGFR or other receptor tyrosine kinases have been one of the major focuses in cancer over these years for precision oncology. They only cover about 29% of all the tumor space. And they've been really exciting because A, you could find them, and B, they were directly targetable. But this other 70+% of the space is riddled with a number of genetic drivers and loss of function mutations that can't be directly targeted through small molecules or other antibody approaches. And we really wanted to look for key vulnerabilities in this space. So to do that, we pursued synthetic lethality. And I guess here I just wanted to step back and kind of give a broad overview of what is that paradigm of synthetic lethality. So in a normal cell, there's normally pairs of genes or networks of genes that can help buffer one another, either through redundancy or other mechanisms, to ensure that the cell, if it's perturbed in any one way, is able to remain viable and progress forward. In cancer, I think, as you understand, there are a lot of genetic alterations that occur to these tumors where they mutate and knock out the genes or alter them, creating them non-functional. And this is a key Achilles heel that we could take advantage of with the synthetic lethal paradigm. So we do screens and other methodologies to find the second gene that is now the Achilles heel, so that in cancer, when we knock out this second gene with a small molecule, as shown on the tumor cell side, we can kill those cells. But in the normal cell, that initial gene is still wild type and buffering the knockout of that gene with a small molecule, and they remain viable. So it really gives us this key therapeutic index from the beginning. And a second really important part, an intrinsic patient selection. So we know the patients we're going after based on the genetic alterations that they have. When we started, there were a lot of different areas of biology that we thought we could go into. But we really decided that focus was important. And you can see based on our initial founders that genome instability was a space that we chose to look at. There were a multitude of reasons for that, but the three main ones were shown here. One, there was precedence from the yeast synthetic gene interaction networks that had already been built in the early 2000s, showing that this is one of three major nodes of interactions within the synthetic lethal space. The second was that genes that are altered and drive genome instability in cancer are usually very early events, so truncal events in the evolution of cancer, meaning we think there would be a chance for a deeper, more durable response as those tumors progress and we are starting to treat late stage tumors. And then last but not least is there's a practicality component to all this, in that there were a lot of druggable enzymes or enzymes that we could go after from a small molecule perspective, which was really the core drug discovery approach that we were taking at Repare. So to go after and identify these novel targets, we have been building this platform that we call the SniperRx platform to identify these novel targets. As I'd mentioned before, it starts with the genetic alteration in mind. So we went through, identified a number of different genetic alterations that are driving genome instability and are overlaid with an unmet medical need in cancer. We then create isogenic pairs of lines in normal cellular backgrounds indicative to the tumor that we're looking at. So a mammary epithelial cell for breast cancer. We have that paired set of cell lines, plus and minus the mutation, BRCA1, for example. And then we come back with a genome-wide proprietary library that we use to screen and identify genes that, when lost, selectively only kill that cell that has the mutation in it and not the wild type. We then take those hits and put them through a number of different validation steps, include looking at endogenous cancer cell lines to identify novel targets that we bring through a heavily structurally enabled drug discovery process to find small molecule inhibitors and bring to patients. Initially, to start, we had picked 16 different genetic alterations, or showing the 4 that we've released to the general public, and that are the underlying components of our key projects that we have progressing to the clinic. All of these were mutually exclusive with one another across the cancer genome when you get rid of the hypermutator tumors, and they represented about 30% of all tumors. And we've been able to actually fill our whole pipeline now with the different outcomes from these different screens that we've done to date. Really ending up in what we have here, which is our furthest along project, an ATR inhibitor that I'll tell you a little bit more about. It wasn't we weren't the first to go after this target, but we felt we were going to be one of the first to go after a really patient-selected approach to using this target, and we'll talk a bit more about that. We also then have a novel target that we know of no one else going after, that's synthetic lethal with cyclin E1 amplification, and the Pol theta inhibitor program that I talked about that we brought in from one of our founders, Aniel Sphere. As well as a number of additional targets. And really the productivity of this approach allowed us to have a very large deal with BMS earlier this year, where they were able to invest in our company, and we'll be providing them with novel targets to be able to bring forward into their pipeline. So I want to step back and talk quickly about 2 of the main targets and approaches that we have within the company. So the first one is cyclin E1. So as I'd mentioned, we're really interested in genome instability and unmet medical need. So cyclin E1, when amplified in cancer, actually drives cells into a shorter G1 and a longer S phase, which results in replication stress and ultimately genetic alterations that lead to genome instability and the evolution of a tumor. In addition, when cyclin E is amplified across tumors, it's associated with a very poor prognosis and high unmet medical need. And I'll talk a bit more about that. And so we created these isogenic lines, did a screen, and we found a novel target that we're bringing to the clinic next year. So cyclin E is amplified across a range of cancers, and its de novo frequency is shown here. We were particularly focused on the uterine, ovarian, and gastroesophageal cancers at the top, not only because of their frequency, but also because of the clear unmet medical need in this space. That is exemplified here in high-grade serous ovarian cancer. So this disease is roughly broken up into two main components. One, where the tumors are homologous recombination deficient. So they have mutations in things like BRCA1 and BRCA2, as well as other homologous recombination genes. And these tumors are extremely sensitive to platinum, followed by PARP inhibitor maintenance therapy. PARP inhibitors being one of the first synthetic lethal paradigms that's made it through the clinic and all the way to the market in patients, showing great success in helping patients not only in high-grade serous ovarian cancer, but also multiple other diseases. However, the other 45% of this disease in green is proficient for homologous recombination and therefore responds extremely poorly to both platinums and the PARPs. And these patients progress very quickly. And this is predominated by cyclin E1 amplification between 15 to 19+% of these patients. So we'd moved, done our screen, identified a target, and this is just showing the single gene follow-up to this particular target in the isogenic pair. So in black is the cell line. This is a fallopian tube cell line to mimic the ovarian cell of origin. It's P53 mutated, again, because this is a permissive mutation that allows cyclin E to actually exist and drive genome instability and is always co-occurrent in ovarian cancer. In black, you can see the control or the two different guides knocking down the gene we identified in the screen, where we have little to no effect, but where you can see when we have cyclin E overexpressed to mimic amplification, we see profound effect on the growth of these cells. One of the key things from doing these genetic interaction screens, of course, is that we're knocking out the whole gene. And as a small molecule biology approach, we always have to think about whether or not that's going to translate to small molecule inhibition of the target. And so in this case, we were able to quickly find key probe compounds that we were able to ask this question. And the results are shown here. So this is the exact same set of cell lines that I showed in the previous slide. Again, a clonogenic assay, but now using pharmacological inhibition of the target. In the wild type in green, no effect. And in the two different clones with cyclin E overexpression to mimic amplification, you can see again, profound impacts on the proliferation and survival of these cells. We were able to move that into endogenous cancer cell line panel. So again, shown here, cancer cell lines that have cyclin E1 amplified versus those that are wild type or a primary cell. And you can see that the ones that have the amplification of cyclin E are hyper responsive to knockout of this target with pharmacological inhibition. And when we brought that into patient-derived xenograft models that have either a low-level amplification of cyclin E at 4x, so 4 copies, versus a larger one with 18 copies, you can see we have 60 to 90% tumor growth inhibition during the dosing period with these earlier probe compounds. We're really excited about pushing this target forward. It's novel. It hasn't been described as being synthetic lethal with cyclin E anywhere, and we cannot find any patent literature or otherwise that says someone has been going after this from a drug discovery perspective within pharma or biotech. And we'll be progressing this project to a drug candidate this year, and in next year we'll be moving into the clinic to be able to test this hypothesis in man. So in addition to thinking about how we could go after cyclin E, when we were early on, we had the POLQ inhibitor. We'd found this cyclin E synthetic lethal target, but we also wanted to think about our portfolio and how we could diversify it, because both of the agents that we had coming up were novel, had never been through animal studies, had never been into man, and so had an intrinsic risk of the unknown unknowns. And we wanted to diversify with a potentially later stage compound that we could progress forward quickly that had a different risk profile. And that was really the ATR inhibition. So ATR up until this point had been pursued by 3 different pharma in the clinic, but none of them had actually gone after a patient selection approach. And it wasn't until actually 2019, so at ASCO of last year, where Bayer for the first time enriched patients with ATM loss, the synthetic lethal interaction for ATR, and demonstrated a really profound impact in that phase 1 study. We also felt there was a rationale for combining and being able to expand out who we could treat. And we thought we could actually come up with a differentiated compound that would help us compete in this space. So to step back briefly, how does ATM and ATR synthetic lethal interaction work? So normally during DNA replication, when replication forks stall, ATR senses those and would normally fix them. When you block that with an ATR inhibitor, they can no longer sense those forks, and the single-stranded breaks can break down into a double-stranded break that in a normal cell is sensed by ATM, allowing that to be fixed and the cell to remain viable. But in a tumor cell where ATM is null, it's no longer able to properly sense those double-stranded breaks. And if even one double-strand breaks goes through into mitosis, it can result in cell death. And it was really this straightforward concept that was tested for the first time by Bayer clinically. And this is just some high-level data shown from their phase 1 and then published actually 2 weeks ago today. And here again, all of the bars with the hashes through them are ATM lost or mutant tumors. You can see only those that had ATM mutations were responding. And the key to this, shown in green in the spider plot, is not only did they respond, but they responded for an extraordinarily long period of time. So this is well over 1 year responses for all of these different individuals, some of them having up to 11 lines of previous therapy and really moving, towards extremely late stage cancer. And they were across different disease settings. So not just within one disease, but was based on the genotype. And this was the data that further drove us to push even harder to drive this target forward and to move into a competitive space. With that in mind, we created RP-3500, our ATR inhibitor, as shown here. It's an extremely potent 20 picomolar Ki, 1 nanomolar for a cell mechanism of action endpoint. It has a great PK profile, low potential for drug interactions, and we initiated our clinical trials in patients in July of 2020 this year. But we didn't want to just rely on asking the same question everyone was doing. We actually wanted to be able to expand out who we could treat and the patients that we thought we could go after. And this was based on that paradigm that I talked about up front, is that synthetic lethal interactions can sometimes be pairs of genes that interact or networks of genes that can interact. And PARP inhibitors, which are used in the clinic to treat normally BRCA1 and 2 mutant patients, also have been shown the ability to actually treat other genetic alterations that cause homologous recombination deficiency. They figured this out clinically, but we wanted to do this preclinically. So we do a process that we call step 2, the SNIPER-RX targeted expansion of patient populations, which is basically a form of a chemical synthetic lethal screen, where we take cells, in this case, up to 8 different cell lines, treat them plus and minus the inhibitor, use our genome-wide CRISPR library to knock out all the genes in the genome, and then overlay that with genes lost in cancer that could be potential biomarkers to pick patients clinically. When we did that, we came up with ATM as our positive control. Again, we knew that these two could interact based on data in the literature and in the clinic. And we found 19 additional genetic alterations that we could go after. These were, of course, hits. They went through in vitro validation, but we also brought them into in vivo, several of them into in vivo validation, with a number of them still ongoing, as this is a very laborious process. But just shown here is an example. So here we take an isogenic tumor xenograft model, one where one of these genes that we identified was wild type, and then the exact same cell where we knocked out this gene of interest. And then we treat with RP-3500, our ATR inhibitor. And as you can see, we have a dose-dependent response only in that cell that has a loss of that gene. And this is the type of data that's given us the belief that we can expand out the patients beyond just ATM, but into other genetic backgrounds, and gives us a competitive edge over some of our larger pharma colleagues that don't know about all of these genes yet, and is the reason why I don't name the genes, as you can imagine, as we're progressing forward. And what did this do when you added these 19 genes? It took a tumor frequency of roughly 2% across all tumors for ATM, shown over here in the blue, and it really expanded that out by 2 to 4-fold across different disease settings, shown in green. This is all the 19 different genes we identified together, looking at their frequencies across cancer, giving us a real opportunity to expand this out. Even if only some of these genes work, we believe we're going to have an ability to, again, treat and get better patient benefit across genotypes. So in addition to thinking about how we approach this, the monotherapy and the patient selection around that, is we were also unsatisfied with combination therapy, going after just patients who were treated with the previous therapy. But we also thought that the PARP inhibitor ATR combination was of particular interest. And this is because PARP inhibitors, when they bind to their target PARP1 in a cell, they actually trap it on the DNA, which basically blocks the DNA replication from occurring and causes replication stress, which activates ATR itself, driving cells into premature mitotic entry, exhaustion, and accelerated cell death if you can block that ATR inhibition. The real problem with delivering this clinically was both of these agents have myelosuppression as their major off-target activity. So they were both hitting this myelosuppression, and it was really hard to figure out how to go after these in a tolerable regimen. So we did a chemical synthetic lethal screen, excuse me, with the combination. And just shown here is an example of one of the genes we found doing this method. Again, in the bluish gray bars are the agents alone or together, or when that gene has been knocked out, one of those ones that we found, again, for this specific to this combination. And as you can see, when you treat, there's very little effect when the wild type gene is there, but when the gene is knocked out in combination, you see a very profound effect on the phenotype. Excuse me. So this is a very rapid and irreversible phenotype that allows us to come in and think differently about the schedule. So treating patients for a shorter duration of time, and it occurs at concentrations of drug that are below that necessary to elicit the maximal single agent activity, meaning we can lower the doses of these agents as well as the duration of exposure, giving us the belief that we're going to be able to get better efficacy with less tolerability issues as we move into the clinic. And with that, I wanted to make sure that we had plenty of time here now for the last 10 minutes to have questions and just highlight what we're moving forward with.
Thank you very much, Mike. So again, if you have questions, please type them into the Q&A box of Zoom. I'm going to ask a question while people do that. So, you know, you mentioned that the Sniper platform uses CRISPR, right, to look at potential synthetic lethal new targets. But there you have a complete loss of function, which, you know, may not be mimicking really drug inhibition. So are you also implementing the new CRISPR tools such as CRISPR-I or others that, you know, may enable you to even siRNA, right? So people are, you know, now dismissing siRNA, but one advantage is that you have a, you know, a loss of function partial rather than a complete inhibition.
Yeah, so absolutely. I think there's multiple components of validation after we find those initial hits from the CRISPR screen to help support and understand this particular issue. So in the one, we go back and do rescue experiments, not only to prove that it was on target to the guide, but we make mutations into the gene that render the catalytic activity or that activity that we think we're going to target, turn it off basically. So if it was a kinase, it would have a mutation that gets rid of the kinase activity, or bromodomain, it would get rid of the binding activity. So we prove that that activity is necessary for the synthetic lethal interaction. So that gives us a bit more confidence. And then as you highlighted in the second part, as we're looking through the cancer cell lines and doing that kind of a panel of cancer cell lines plus and minus our biomarker, we often use either RNAi technologies or CRISPR-I, depending on how robustly that knockdown is across there. Many of the reagents end up knocking down the target completely, so we have to do a lot of work around that. But we use those two different methods to have partial knockdown as well as a rescue of the enzymatic function to prove it's part of the synthetic lethal interaction. And then I think the and the last part, just to highlight there, if we're really excited about a target, it makes a ton of biological sense and the validation is looking good, we move as quickly as we can to pharmacology. Because at the end of the day, we still want to understand and make sure that the pharmacology actually is impacting the same way that the genetic alterations are.
Thank you, yeah. So a question relates to synthetic lethality platforms across companies, right? So a lot of companies have now adopted this principle, I guess, in a new way to look for potential targets. So can you comment on the differentiator of your maybe technology platform compared to the other ones, and now you think you may have an advantage?
Absolutely, and I'll give the key caveat up front is that I unfortunately don't have all the insights into what exactly everyone else is doing. I'd love to. If anyone else does, that would be great. But I can do what I think I understand from public presentations and other approaches. So I think one of the major approaches that was done in academia and in several of the biotechs was using large cancer cell line panels. The Sanger 500 panel, the Broad, I think KSQ and others have taken this approach. We think it's a really powerful approach, but what we found is that actually the number of false negatives is actually quite significant. So CRISPR is a lot more about false negatives, whereas RNAi was really all about false positives. And that's because every one of those cells has different defects in the DNA damage response and how they fix the DNA after it's been cleaved with a CRISPR reagent, meaning that in one cell you could have 80% out of frame, whereas the next cell you might have 50% out of frame. And then when you have only 2 or 3 cell lines with your biomarker, you may actually just completely miss a signal. So because of that approach, we wanted to start with a very clean, normal cellular background. So we have over 10 different cellular backgrounds that represent the different cells of lineages for, mammary epithelial cell, fallopian tube line, et cetera. It's taken quite some time to get these up. They're immortalized, usually with HTERT if possible. So we have a really clean background, and they've been optimized to ensure that the CRISPR-Cas9 is as efficient as possible. So we can recall 98% of all the essential genes every single time we do the experiment. We believe minimizing our false negative rate. And one of the proofs of that principle was we pull out PARP1 for BRCA1 and BRCA2, whereas you would never see that in the other panels. And then the other is a practical component. We started this roughly 5 years ago in Dan DeRoche's lab and have been building this up. And so we completed those 16 screens some time ago. We're never under the illusion that academic or other competitive industrial labs won't find equally interesting targets than the ones that we've looked at. And so executing the key component of that, both internally, and one of the main reasons that we did the deal with BMS is that we didn't want targets sitting on the shelf that we couldn't go after because of our own bandwidth, just to let somebody else come out with it in the literature and then let the whole world go after it. So we want to keep that competitive edge that we think we have from starting early and progressing forward.
Very nice. Another question relates to, often you see a lot of resistance, therapeutic resistance linked to targeted therapies. And is there any evidence that employing a synthetic lethal strategy may overcome this problem? Or has drug resistance also been seen for the known, synthetic lethal combination? I'm not aware of PARP inhibitor drug resistance, but can you comment on that maybe? Yeah.
So I think drug resistance is probably inevitable in cancer, unfortunately. Cancer always finds a way. And so far, PARP is the best example of a synthetic lethal target that's been around and has enough to understand that. And so in there were 2 things that showed up. One was mutations directly in BRCA1 or 2 that put it back in frame and make it a functional protein again. So now they're HR proficient and the cells no longer responds to PARP. There also have been other alterations in that homologous recombination pathway that, again, bring those cells back to being HR proficient, and therefore they will no longer respond. One of the reasons why we thought the ATM-ATR interaction was of most interest was that ATM is not only important for the initiation of a tumor, it's also necessary for the maintenance of a tumor. So you can't fix it later on without killing the cell. So it takes away at least one of the major modes of resistance outside of direct point mutations. But it's unclear for that mechanism how we would be able to approach resistance at this point, and something that we're looking into internally and in collaboration.
Good. And maybe one last question before we move on to the panel discussion, but you also mentioned that during your talk that the ATR inhibitor is going to be most likely a best in class, not the first in class. So the question is about what goes on in the strategic decision within a biotech to, I guess, pursue this. Can you comment on this? Pursue this strategy when you know that there's another company that has a similar inhibitor.
Yeah, no, absolutely. So I'd say that there's a whole number of things that come into it, but I think our main focus was 2 things. One is we knew that ATM and ATR, there was literature that highlighted a synthetic lethal interaction in there, but there was the fact that no one was actually going after it clinically. Everyone was doing combinations or just going after chemo combos. It wasn't ever selecting for patients. So that was one. The second was we did an ATM screen, and again, the ATR pathway came out as a strong hit. So it gave us more belief that we should go after this. And it helped diversify our risk as a company. Complete novelty, not knowing what would be happening as we moved into later stages of the drug discovery process in early clinical, we wanted to be able to balance out those risks. So in this case, the risk was really competition. We thought we could create a competitive molecule, if not a best in class. That's something that we would have to see in the clinic, obviously. But that's why we also then looked for novel patient selection biomarkers that we could go after and continue to try to expand out the biology innovation. But the only other part I would highlight is just, I can't count the number of times where the first molecule or the second molecule wasn't the one that got to the market. Because every molecule is slightly different, has a different PK profile, or has a different toxicity profile that may be due to activities that just aren't understood. And so I don't think the game is ever over until you're on the market and you're competing. And so it's really those different pieces that come together that gives us a belief we have a real chance to help save patients' lives and to really push that forward.